we can start. So before we begin, I would like to acknowledge and pay respect to the traditional owners of the land on which we meet, the Gadigal people of the Eora Nation. It is upon their ancestral land that the University of Sydney is built. I would like to pay my respect to the elders past and present. Welcome to the Sydney Nano Distinguished Lecture on Cancer and Nanomedicine. It's my great pleasure to welcome Distinguished Lecturer Professor Maria Kavallaris to the University of Sydney. She is truly a rock star in Australian innovation and research. Maria is the founding director of the Australian Centre for Nanomedicine, chief investigator on ARC Centre of Excellence in Convergent Bio-Nano Science and Technology at UNSW, and NHMRC Principal Research Fellow and head of the Translational Cancer Nanomedicine team at Cancer Research Institute. She is internationally recognized for her research in cancer biology and therapeutic. Her research and leadership have been recognized through basically many times, just to name a few, through Australian Museum Eureka Prize and being named by the NHMRC as the Australian High Achiever in Health and Medical Research. Maria is the Fellow of Australian Academy of Health and Medical Sciences and a Fellow of Royal Society of New South Wales. And in 2019, she was appointed a member of Order of Australia for her significant service to medicine and medical research. With that, I would like all of us to welcome Maria here and thank for accepting our invitation.
Thank you. Thank you, Amit. It's actually a great pleasure to be here, and thank you for the invitation to come along and tell you a little bit about the work that we're doing. I was told you're a fairly broad audience, so please forgive me if I start off with some really, maybe for some people, very basic things, but then work up to what we're doing. And, you know, as I'm actually a cancer biologist, so don't ask me any really bad or heavy-duty chemistry questions. I'm actually a cancer biologist by training, but I've been working in the nano field since 2005. And it's been a great way to integrate multidisciplinary research into the types of things we want to do. And my focus is very much on the application of nanotechnology to medicine. Just a very quick, some disclosures, because I'll be showing some data on Inventia and the Engenic work that we did as well. So I don't have to tell this audience that the scale of nanoparticles. You know, we often say to people, nanometers is 1,000th the width of a human hair. I'm a little bit freaked out that the fly here is much bigger than the 4-year-old child, but this is what the figure that the conversation picked to put in the article that we wrote about. It was an opinion piece about nanomedicine and applications for childhood cancer treatment. So people often ask me, what is nanomedicine? And we define it as, I guess, the engineered materials at the nanometer scale that we're developing for medical applications and other people are developing for medical applications. But it truly is, an interdisciplinary type field. As many of you know, and I probably don't have everything here, it does cover things from chemistry, engineering to physics, biomedical research, medicine, biology. These all, when they come together, can really achieve some great things together. And that's really when we set up the Australian Centre for Nanomedicine, we had in mind to approach, nanomedicine from the disease perspective. So what is the disease problem and how can we use nanotechnology to help come up with good solutions? And the sort of things that we work on in the centre, and some of this I'll be presenting today, is in the area of diagnostics, therapeutics, and also theragnostics, the combination of being able to detect disease or detect systems and also treat at the same time. And this is like I said, I won't go through all the part. I'll be focusing a lot on therapeutic delivery, but also some work we're doing sort of on diagnostics and predicting therapy as well. So my talk will be focused on cancer, and as many of you know, I think if I can see a hand of someone who has not either directly been impacted by cancer or doesn't know someone by cancer who's had cancer, please put your hand up now. And this is very common in any talk we give, and I've given talks to 2,000 people at a time, not seen a single hand go up, because cancer impacts everyone's life in one way or another. And the latest statistics say, because we're living longer as well, that almost 1 in 2 people will be diagnosed with cancer in their lifetime. So just to think about the degree of the problem, you know, we had 18 million cases in 2018. In 10 years' time, this is going to go to 20 million, and this will result in 13 million deaths. And just in the hour we'll be sitting here, you know, there'll be like close to 2,000 people will die from their disease. So it's a major global problem. Now, cancer is a complex disease, but people say, well, you know, there's all this money going to research, cancer research, and I know other disciplines sometimes, you know, it's the envy and say, why are you spending all this money on cancer research? It's because it's a highly complex disease, and it's not one disease. That's the thing to remember. So even within a single patient, if they're diagnosed, for example, with metastatic disease, just to say this colon cancer was the primary tumour, and they've got metastasis to the brain, they've got metastasis to the lung. Even within these tumours, you can see the little blue cells, they're going to be common to the tumours, but you'll also get variation, so you get heterogeneity within those tumours. That's why sometimes you hear a patient is treated for cancer, they have surgery, they have chemotherapy, they're doing really well, the tumours have shrunk, and then 3-6 months later, sometimes a year later, the tumour comes back with vengeance. And then when you try and treat those patients, you know, they're not responding to the therapy as well. And it's because what you're doing is that you're knocking out some cells, but you're getting this heterogeneity. So as you go through each treatment, you're selecting out these resistant populations of cells that are able to bypass the treatment. So we really need to be thinking about how can we improve therapy, how can we make therapy better, how can we make therapy more effective, particularly at the early stages. So early diagnosis, prevention is probably the best thing you can do, but we know we can't prevent. 85% of cancers are environmental in origin. So then early detection is incredibly important, because the earlier you can catch a cancer, the more curable it is. And then the next stage is, but we know a lot of cancers are already metastatic at diagnosis. We need to also have better treatments for those patients. So we asked the question, you know, how can we use nanomedicine to influence how cancer patients are treated? So what I'll be covering a bit in my talk today is ways that we're developing to identify the most effective treatments, developing strategies to deliver chemotherapeutic payloads to tumours, and also targeting undruggable genes in tumour cells as well. So one of the key questions is, how can we identify the most effective treatment really in the most timely, accurate and predictive manner? And this is, as I said before, it's quite important in terms of not only to improve patient outcomes, so you try and give them best treatment early, but also to avoid the use of ineffective therapy. Many therapies, even ones that don't have a lot of toxicity, have side effects. So if you can avoid ineffective therapy, that's incredibly important. Our institute started a cancer precision medicine program about 5 years ago, but many other groups are also doing this. Ours is in childhood cancer, but it was originally started in adult cancers. And essentially what happens is that when a patient comes in, the pathologist takes a sample, they do their profiling to look at what type of tumour they think it is, and then there's molecular profile where they sequence the tumour to identify potential features of that tumour that may have what we call targetable mutations. In other words, there's a drug that targets those mutations or those alterations. And the idea is to treat the patients. When they relapse, try and do that again, find out what the changes are and treat again to try and increase that. in adult cancers, it hasn't been such a fantastic success story. I mean, probably the best success story, and that's been around much longer than these type of profiling, is probably in breast cancer, where people find the BRCA mutation or the HER2 alterations, where they're HER2 expressing tumours, where there's targeted therapy. But in general, it's not been as successful. And I've got less than 50%, but in adult cancers, it's about less than 20%. We're now finding in our childhood cancer program, we're taking the 30% of children that have got the worst prognosis cancers. These are kids essentially on death row. The chances of survival is very small. So we're focusing on those children because that's where we feel we can make the biggest impact initially. And in our case, we're actually finding that around 60% of kids, you can find these actionable mutations, which is quite exciting. Getting access to the drugs is another story, and give me a few drinks and I'll tell you about that. The other thing we do, and other groups do as well, is if there's sufficient sample, you also try and grow them into patient-derived xenografts in immunosuppressed mice. And this is a way of expanding the material and trying to keep it as close to the human condition as you can. And there's pros and cons with this approach, but probably the biggest obstacle, and the idea is you then can treat the mice with different drugs and different combinations to see what might work. The biggest obstacle here is that tumours can take a long time to grow, or they may not grow at all. And it's also incredibly expensive to do this type of work. And where we do get enough material, we actually try and grow the tumour cells in culture. And we know that cell-based drug screening can be, quite a useful tool, but more and more we're finding that, growing cells on plastic is not highly representative. And we know that genetically, if you compare cells growing on plastic compared to growing in 3D spheroids to a human, the patient tumour, the genetics of the 3D grown spheroids are much, much closer to the human tumour than the two-dimensional grown cells. So the reason for this is because in a tumour, you have a very complex environment. So it's not just the tumour. We think of the tumours and we talk about treating a tumour, but we are not just treating tumour cells. We've got, tumour cells are surrounded by quite a complex environment that they recruit to that tumour. And they stimulate recruitment to that tumour because they release different factors. You've got the blood vessels, immune cells, you've got fibroblasts, the endothelial cells. There's a whole lot of cells in there that not only is the tumour helping to stimulate to be there, but they're also helping the tumour grow and spread. So we need to sort of have good models to try and replicate these type of systems so we can better understand what's going on. So traditionally, if you want to grow 3D spheroid models, you grow them on low adherent plates, and the cells like to be in contact with something. So that's why if you're doing tissue culture, they adhere to the plastic. But when they can't adhere to the plastic, they will adhere to each other, and they will form these spheroids. And you can see this little movie here where, we show that you've got a single cell suspension, and then the cells come together, they get attracted to each other. And then, and if you actually look at these spheroids, you have a zone, what we call the proliferating outer cells, the quiescent cells, and the necrotic core. This is actually what also happens in a human tumour. When it gets to a certain size, the outer part of that tumour is proliferating. You've got some cells that are not really dividing that much, not doing much. And then in the centre, you'll get necrosis, and because you get these hypoxic regions in the centre. And so that's what we're trying to replicate here. But there's a number of challenges, right? So if you want to try and mimic the environment where you've got extracellular matrix, etc., traditionally what we've done in the past is, we'll grow those 3D spheroids and we'll place them onto a matrix, and then look at their ability to spread out from those tumors, from those spheroids, those tumor spheroids. And we can measure the distances, etc., and analyse that. Or if we really want to look at metastatic spread and how these tumours are moving in a complex matrix where they have to use proteases and digest that environment, we can actually embed them in a matrix, and you can see this outgrowth that happens. One of the things with this is that it is a closer mimic of what's going on, and it actually allows you to see how those tumour cells are behaving. I mean, in both these studies, we were actually manipulating genes that we thought were involved in metastatic spread, and being able to show through these approaches. And then, through this system, you have the matrix interactions, and also the passage of nutrients, etc., through spheroid and matrix, a little bit closer to the human situation. The big problem is that it's slow, labour intensive, and has low reproducibility because you're doing everything manually. And the other thing is it costs you in a lot of postdoc lives, because the postdocs, when you tell them to do this, they absolutely hate it because, to get these images, to get this and prevent the spheroids from floating to the top, which you can't do, and being embedded, it probably takes between doing 20 to 50 of these spheroids to get them right. So one of the things we were really interested in doing is understanding how can we produce these spheroids to use in downstream applications in a reliable and reproducible manner. And we teamed up with my close collaborator and co-director of the Australian Centre for Nanomedicine, Justin Gooding, and Julio Ribeiro, who heads the startup company Inventia Life Sciences. And what you can see here is a 3D bioprinter we were part of the development for, and I won't go through the full development of the engineering of the printer, but last year this won the Good Design Award for its category, but the Good Design Award, Industry Award overall last year. So that was very exciting, and I'm going to talk to you a little bit about some of the biology and how we validated it. So this is how it works. So for this one, this is a particular application where we're making a cup. So basically we've got the bioinks, the activator, and it sets as you go along. And what we can do is once we form the cup, we can drop the cells inside the cup. And these cells, because this particular gel is an alginate-based gel, so they don't adhere to the side, they form these 3D spheroids. And we can do this like in a high throughput format. When we do this, so if you take an image of one of those cups, this is the little rosette that looks a little bit like a flower, but that's actually the spheroid in there that's actually formed. So, and you can actually do downstream. You can either look at different measures in here, like cell death, cell proliferation, or you can also extract them and do other downstream applications as well. And what you can see here is different. We've done quite a range of cell lines now, and the company's done even more. These are some neuroblastoma cells, lung cancer, and glioblastoma, which is a brain tumour. And what you can see is that as we add a set number of cells, you can monitor the growth of that spheroid over time. And same with, you can see glioblastoma and the lung cancer cells. Or alternatively, we can actually adjust the number of cells, so the number of droplets we put in there, which contain different amounts of cells ranging from 9,500 cells in 2 droplets up to 33,000 cells in 7 droplets. And then this is a set period of time, and you can see you can adjust the size of these spheroids as well as they grow. But they're still forming the spheroids in a similar manner that I was showing earlier. And we actually needed, when we're optimizing and developing this technology, we had to also prove that these were consistent with what manually was being used. The big difference here is this: we wanted something that we could do reproducibly in high throughput, so we can do a whole lot of stuff. And what we were able to show, we did some lattice light sheet microscopy to actually look at, are these spheroids compacting in the same way? Is the bioprinting affecting them in any way? And we stained them with actin and the Hirsch stain, and what we were able to do was measure the distances between the nuclei. And what you can see is the manual and the bioprinted are virtually identical. We also looked at the proliferation marker, Ki67, in these spheroids, and these are different layers of the spheroids. We're able to show that they were very similar. So that means whatever we're producing will be compatible with what's been made manually. Also the hypoxic properties, like the centre of the cells, etc. We measured HIF1 alpha in these spheroids, and They were very similar. And also the stem-like properties, because when you make spheroids, now I should just, a terminology thing I'll mention here. When I'm referring to spheroids, it means we're using cancer cell lines. Later I'll be talking about tumoroids, and that's ones that we develop directly from patient material, patient-derived material, okay? So in this case, we're using a pure cell population of cancer cells. And this is just, when we take the spheroids, you can actually take the spheroids out, embed them and section them, and actually look at the, the pathology of the spheroid. So we had a pathologist look at this and who said it was consistent with other tumours he'd seen in the mice, but these were grown ex vivo, obviously. And this is a dose response to doxorubicin of these. This is neuroblastoma cell line, and you can see that with increasing dose of drug, we can get a really nice dose response happening. And we've done this with a range of drugs now to show that we can do that. But ultimately, it's not cell lines we want to be using. We really are interested in patient samples. And I've already mentioned that patient samples grow in a complex environment. And so one of the things we want to be able to do is extract from a tumour and bioprint, and then analyse these and look at, make sure, so we're in the process of actually, confirming that genomically and phenotypically these behave very similarly. So what I've got here is some patient-derived xenografts. So because the samples are so precious from the patients, we've got an unlimited source of these patient-derived xenografts, which are growing in the mice. They've been validated to be genetically very close to the original patient sample. So what we did is we took some of these, and first we optimised the conditions for growing them manually, which is not an easy task because they don't like to sort of grow manually. So these are grown as spheroids in suspension, and again, we did the histology on these spheroids, and the pathologist confirmed that these look very similar to the original tumours. But what was very exciting is that we were actually able to bioprint these primary samples. So we're taking a soup of these cells, okay? So we're counting the cells, but within those cells, we've got tumour cells, we've got some other connective tissue that would be derived from the mouse, and the tumour itself. So, and we bioprint those, and we're able to get a dose response again with those cells. So the main concern for us is that primary tumour samples are very sensitive to cell death. So we were concerned that the bioprinting might affect that, but what we found was it didn't. So that was very exciting. And then we also wanted to see was, can we actually start predicting like responses based on what the mouse responses to the drugs were initially? Because obviously, again, we have to do this initially in mouse models of the patient samples, and then we'll move to clinical samples. But basically, we had one sample that was highly sensitive to the drug temozolomide. It's a neuroblastoma sample. So in the mice, it was highly responsive, and it actually had complete response. So some of the mice were treated, the tumours either disappeared or they were incredibly small. And here, or they stopped growing completely. And this sample was one that had progressive disease. That means the mice were being treated, but the tumour just kept growing through it. And what we found, and it's quite exciting, was that we needed a lot less drug to inhibit the growth of these spheroids, the bioprinted spheroids, compared to the resistant sample. So we've just been funded by Cancer Australia for 3 years to now build up this work even further and actually move to patient samples as well, and do all the comparisons of the genomics, etc., of the primary tumours. So we're quite excited about that. So I think, combining molecular profiling with phenotypic profiling and drug responses is a powerful approach for individual patients. And we're working very closely with clinicians and the Zero Childhood Cancer Program to progress this work. So I'll just introduce you a bit more to childhood cancer, because a lot of people say, well, why do you work on childhood cancer? Why there's all this work done on adult cancers, can't you just transfer all of that? And the answer is no. And it's because the biology of childhood cancers is very, very different. The types of cancers the children get compared to what adults get vary dramatically. And the biology of those is very different. While adult cancers have a lot of mutations, childhood cancers have very few mutations. They have translocations, but they don't have mutations. So childhood cancer is responsible for the greatest number of deaths of children in developed or high-income countries. And worldwide, there's just under 6,000 kids diagnosed each year. Now, it's been one of those success stories because 80% of kids actually survive childhood cancer. But for some childhood cancers, there's actually been no real progress. So that disease, neuroblastoma, I was showing you, less than 50% of the kids with the aggressive form of disease actually survive. And if you look at low to medium income countries, the survival rates are dreadful. And toxicity remains a major issue. The majority of kids are treated with chemotherapy because unlike adult cancers, where, you've heard a lot about immunotherapy for cancer, it's not as common to use immunotherapy for children. There's a couple of things on the market, but it's not the same as adult cancer in terms of immunogenicity. So, because kids are given systemic chemotherapy, it goes all around their body, and it doesn't only affect their cancer cells, but also the normal healthy cells. And short term, the side effects can be quite severe. So while we're sitting here now, 50% of the kids in any children's oncology ward in Australia are being treated for the side effects of their treatment, not their actual treatment. So these are treatments that are meant to save their lives. And they do save a lot of lives. But, many need, hospitalisation. The other thing is for the survivors who have had aggressive cancers and been treated very aggressively, that means, high-dose chemotherapy, bone marrow transplants, radiation, etc., or even some of the brain tumours, there can be a lot of long-term side effects. And we're only realising now, I think it's been estimated something like 70% of childhood cancer survivors have some level of side effects, with 30% of them being quite severe, like cardiac problems, fertility, endocrine problems. So there really is an urgent need to really be thinking about effective and less toxic therapies for childhood cancer, and hence my interest in this space. I've already mentioned neuroblastoma, and this is the most common extracranial, non-brain cancer, which is a nerve cancer. It arises in the sympathetic nervous system. The problem is that it often starts abdominally. And so 70% of the kids at the time of diagnosis have got metastatic disease. So that's very high. And advanced stage disease, and these are kids with particular genetic alterations and late stage, are rarely cured. And the ones that do, like I said, have long-term side effects. So we teamed up with a company called Engenic that developed a bacterial-based nano delivery system. So this is where these mutated bacteria bud off a very distinct spheroid system that's got a membrane, but it's got no genetic material in the centre because there's no nucleus. And they can flush these out of lipopolysaccharide as much as possible, and they can load them up either with drugs or siRNA. So these 400 nanometer spheres, when I first saw this work, I thought, how is a 400 nanometer particle going to get into tumours? Because it's we normally say 100 or less is better. But what they did is they genetically engineered these bacterial nanocells to actually have with bispecific antibodies on the surface that will recognise tumour cells. So we thought, well, look, it had gone into clinical trial in adults where they just took terminal patients and tested them out. And we were keen to see whether it would be effective in neuroblastoma. So the marker we were going after, the antibody marker that they had that was quite effective, was this epidermal growth factor receptor. And we screened a number of our neuroblastoma cell lines and identified one that had very high levels, this BE2 cells, and also one that had very low levels of the receptor. And we used that for subsequent studies. So what we've got here is we grew the cells, and I'm showing you an abbreviated version of this work, but we grew the cells as 3D spheroids, and we wanted to see, because doxorubicin is the drug that they use to load the nanocells, it's fluorescent, so it's quite conveniently fluorescent, so we can see it. So if you use doxorubicin alone, it usually only penetrates the first cell layer of a 3D spheroid. I can see it 1 hour and 4 hours here. When you use the this EDV, it's called an ingenuity delivery vehicle, but for convenience, I'll call it the nanocell loaded with doxorubicin, but no antibody. You can actually see it actually seems to penetrate quite well, which is quite surprising. And we don't know if it's because of the sort of like bacterial membrane that it is on these nanocells. And then if you've got the antibody attached to this nanocell with the dox loading, you can see it's very similar. And we were able to quantitate this and show that there's actually no difference between these two. And this was sort of quite a surprise, and the company said, no, they shouldn't be, getting in there without the antibody. So we don't quite understand how that works. But when we actually look at efficacy, that means that we're looking at cell death in these spheroids. This is just the empty nanocell dox. Now, the dox in all these studies was used at 100-fold greater concentration than what we were delivering in the nanocells. You can see that the EDV dox was very similar to the dox here. But then when we had the antibody attached, it actually had a much greater effect. We also checked the antibody alone, and it didn't actually influence the survival of the cells. So it looked like, even though it was penetrating these spheroids just as effectively, it was a bit more effective when you had the antibody combined. And then when we went into mice with this, you can actually see here the control is this blue line here. The purple is the nanocell with the loaded with dox and the antibody. And you can see that the most effective was the actual one with the antibody loaded onto it. And also you can see here, this is, we excised the tumours, and you can see that these were very small, and indeed some of the tumours actually disappeared completely, and we couldn't actually measure them. So it was actually quite effective. And when we did some survival curves, we found again that we could prolong survival in the mice. Now, we only tried a limited number of just one type of treatment schedule, but it would have been good to have done a bit more. But we published this work a couple of years ago, and as a result of that, one of the clinicians that I work with also took this to clinical trial, just changed the drug loading that is being used. So the trial was meant to finish late last year, but it looks like it'll be winding up early this year. Again, this is in kids with that express, their tumours express EGFR, and that have got high level, have got high levels of EGFR as determined by a cutoff that we had. And so far, it's mainly central nervous system tumours where that's happened, and brain tumours. So. All right, so I'm just going to switch gears now. After, it's just one of the delivery systems. We've worked on a number of delivery systems, but that was the only bacterial one we did. The other ones are engineered materials. The next thing we wanted to do is look at whether we can silence cancer genes, drivers of either cancer growth or drug resistance. And not all genes are easily druggable. It's not always easy, particularly if there's a lot of homology to genes that are not causing disease as well, related proteins. And we used an RNAi approach, or RNA interference approach, which I'm sure all of you have probably heard of. But this is an evolutionary conserved mechanism that initially identified in plants and insects, etc., that can actually control gene expression. And you can make synthetic short interfering RNAs that to use for gene silencing. And we do this routinely in the lab for various types of things. And, this is not magic. I mean, there's a lot of clinical trials underway to try and using siRNA or other types of gene interfering to block genes that are involved in cancer. But these are clinical trials, and it wasn't till just over a year ago that the first siRNA nanotherapeutic, it's a liposomal formulation, was actually approved by the FDA, and this is for a rare form of amyloidosis, which is amyloid buildup in the liver. And patients, and this is terminal, so once it starts happening in these patients that have inherited it, they lose their liver unless they can have a transplant. So this is very exciting. We've been waiting for this for a long time, but there's quite a few clinical trials on and development of siRNA therapeutics. So our idea was, if you're going to go after a gene that you want to silence in cancer cells, you want to go after a gene that has multiple targets in those cancer cells. And we've been working for some time on a particular cytoskeletal protein that actually impacts cell growth, metabolism in the cells. It's a drug resistance gene as well. It's also involved in invasion and metastasis. This gene is called TUB-B3, which encodes for beta-3 tubulin, and it's not considered druggable because it belongs to a family of proteins, some of which have really important normal functions in tumor in cells as well. So, in terms of going after for siRNA, one of the big challenges is that naked siRNA cannot enter cells. And indeed, if you gave it systemically, it will be toxic and probably form an immune reaction as well. And it also would degrade rapidly. So you do need a way to deliver it. So the siRNA is stable, you can give clinically relevant doses, it's non-toxic, and it's tissue specific. So, and that's when we started working on nanoparticles for delivery of siRNA. And the nanoparticle we're working on this with Tom Davis's group when he was at UNSW, but also we still work on these type of systems. We started off with something called Star-Nanoparticle, which is a polymeric system synthesized by Raft polymerization. And we've got the PDMA, which is via electrostatic interactions can be complex with the siRNA, and the PEG to actually shield the siRNA as well. And we started developing this for pancreatic cancer, which is it's a lethal cancer. Less than 5% of people are alive within 5 years. And part of the problem is it's got all this pink stroma around it. These tumour cells are in blue, but the stroma prevents drugs from getting in there. It's not very vascular. So we're looking for something that was small in size, relatively small in size, and its surface charge was as close to neutral as possible to see if we could penetrate these tumours. And what you can see here is this is just some fluorescent siRNA in the Star nanoparticles compared to the naked siRNA, and you can see there was good accumulation within the pancreatic tumours of these nanoparticles, even though, like I said, they don't have they're not very vascular. And then the red fluorescence here is a frozen section of these tumours showing that the siRNA is getting into these tumours. And we also did some staining of the blood vessels, and you can see that the Star siRNA was actually able to extravasate from these blood vessels to get into the surrounding tissue. And then when we treated the mice, and this work was all done, I should point out, with Phoebe Phillips at the Adult Cancer Centre at UNSW, who's an expert on pancreatic cancer. And what you can see here is that when we took the tumours out, we could see a profound knockdown at the gene level of this beta-3 tubulin in these tumours. And these tumours, by the way, have the stromal components derived from patients included in these orthotopic tumours. And we got, if you look at the protein level, you got really nice knockdown by the immunohistochemistry as well. So, so that was very exciting, and there's still another paper coming out on that work, a follow-up paper for combination therapies. But the other cancer we were really interested in was a cancer called medulloblastoma, which is the most common brain cancer in children. Now, it's actually, there's 4 subtypes of medulloblastoma, and I won't go through them. 2 of the subtypes are quite treatable and they respond really well. The other 2 subtypes do not respond as well, so kids need high-dose chemotherapy. But unfortunately, the kids also need major surgery. So even if they've got the good subtypes and they survive, they often have cognitive issues and other type of problems as well. So we really want, and the more aggressive tumours are really chemo-resistant. So we wanted to work a little bit further on these star nanoparticles to see if we could make them a little bit smaller, because trying to cross the blood-brain barrier is probably the next holy grail in cancer medicine. And so we worked with Tom Amadeus's group again, and there was only some minor tweaks in the formulation, but we're actually able to get these. The previous ones I showed you for pancreatic cancer were 35 nanometer particles. These ones, when they're complex with the siRNA, are actually 19 nanometer particles. So we're quite excited we could get these a little bit smaller and also quite uniform. And you can see here, this is work done by Helen, who's just submitted her PhD, and she's actually showing here that the star nanoparticles can deliver siRNA to inside the actual medulloblastoma cells, and she's just got lipofectamine there, which is a reagent we use in the laboratory as a positive control. She also went on to actually understand how these particles enter the cells and actually getting released. Just in the interest of time, I can't go through a lot of this. This is not published data, but she did some really lovely work showing, you expect that the siRNA comes in with the nanoparticle as the pH changes within the cytoplasmic compartment, and it's slightly acidic in the endosomes, you get rupture and release of the siRNA. And what she was able to show, you want this to happen in the early endosomes, because if it happens in the late endosomes, they might get targeted off for degradation. You won't get silencing. And what she was able to show, she stained in green here the early endosomes, and she's already starting to see release of the siRNA out of the endosomes. And that's an endosome that still has it in there, but and also show that there was knockdown of the gene she was looking at there. So that was quite exciting. And she started working on Polymer-like kinase 1 as the target gene. This is another gene that has multiple targets. Not only does it affect cell growth, cell proliferation by arresting the cell cycle, it also causes DNA damage and cell death. And it's upregulated in many cancers, including medulloblastoma. It's actually a poor prognostic marker. And you can see here she was able to show with her star nanoparticles, and this is being done in cell lines at the moment, that she's got a DNA damage response, as indicated by the gamma H2AX staining. You can see a massive increase in the cells treated with the PLK1, and also, phosphohistone H3 is a cell cycle marker, and again, you're seeing cell cycle arrest and also cell death in these samples, so apoptosis. So you can see very few live cells remaining, and actually she has to pick a time point quite early to even find some live cells, because it really they become very sparse and die off within 72 hours. And you can see increased PARP cleavage, again, apoptotic marker there. So having validated the target in these cells, we then wanted to know whether we'd be able to cross the blood-brain barrier, because, again, something that is really important to us. So we moved across to a zebrafish model, and we collaborated with Ben Hogan, who was originally at UQ and is now at the Peter MacCallum Cancer Centre. And we're able to, Helen went down and they did this posterior cardinal vein injection of the fluorescent siRNA. So you can see a little bit at the site of injection. But what was very exciting is that they could actually see accumulation in the brain. And these are more high-resolution images. This is the siRNA alone, and the green is the actual blood vessels. So Ben's expertise is in vascular research. So he has this zebrafish model that's is screen fluorescent protein that stains up the endothelial cells. So every single endothelial cell in this zebrafish embryo is lit up. And you can see that, we've got the merged images. You're not seeing any siRNA. And then here you're seeing, you're actually seeing the Cy5 siRNA. And what was exciting is not only do we see it at the blood vessels, we're seeing it coming out of the blood vessels as well. So that was very exciting. And they've done a bit more analysis on that. So Helen's been able to show that, we can target with these Star nanoparticles, at least in vitro, and the Star siRNA is getting internalized, and it's causing cell death, and that's the main thing. What we're doing now, we actually have data now in mice to show that the Star siRNA does get into the brain tissue, and we've got some preliminary data that shows that it crosses the blood-brain barrier, and I didn't update the slide. It's work we want to repeat. That's why I'm not showing the data, just to be 100% sure it's working. But she's also taken some frozen sections of the orthotopic medulloblastoma tumours in the mice, and it appears that there is some siRNA in there. So the next phase of the experiments, and she's staying on to work with us for this year to actually finish off the work. The next phase now is to look at, validate that it is indeed going to the brain tumours themselves, but also to look at efficacy studies in these mice as well, because ultimately we want to do that. If we don't see as much efficacy as we want, we're also planning on other routes of delivery. So we'll also try direct tumour injection intrathecally, and we're trying some other methods like being able to inhale the siRNA as well, because you can transfer through the olfactory system siRNA. But in the first instance, we'll just see the traditional method, because it does seem to get across. What the challenges are is, how frequently you should dose the mice, because siRNA and the nanoparticles will have a finite time circulating. So this is ultimately what we'd like to do, go after some of these really difficult to treat cancers. There's more difficult cancers than medulloblastoma, believe it or not. There's one called DIPG, which is diffuse pontine glioma. Fortunately, it's rare. Unfortunately, it's 100% lethal. So most children die within 9 months, and the tumour grows down here. So we're doing some work to look at ways of doing that, looking at ways to improve the blood-brain, to open up the blood-brain barrier a bit more, to get more material. But with the medulloblastoma cell work, we also know that if we combine that PLK1 with chemotherapy, and low-dose chemotherapy, it's a really great combination. So ultimately, that's what I want to do. But I just want to leave you with just one last message, is that I think cancer delivery is very complex, and one size does not fit all. I started the talk by actually talking to you about the complexities of tumours and the tumour microenvironment. And depending on the type of tumour you have, the location of it, the environment those tumour cells are growing in will be different for these different types of tumours. And even within the same patient, there'll be differences. So there's still a lot of challenges, but I think there's also a lot of opportunities. And we need to be thinking about how can we treat people more effectively? How can we, we do that with a lot less toxicity than what we have. And I'll just wrap up by, I'm not going to go through all my collaborators, just some of the main people. Particularly want to thank Tom Davis, who Monash, but now moving to UQ. It's all very complicated. He's 20% Monash, 80% UQ. And Josh McCarroll, who I've worked with very closely for years on this, particularly Star nanoparticles, and also Phoebe Phillips in the adult cancer medicine program as well, plus like Marley and Moon's son, who've worked on the 3D bioprinting, and our clinicians and the patients who allow us to get access to these samples as well. So thank you.
Okay, I think we have time for a couple of questions. Thank you for very great, impactful research you're doing. Yep.
Thanks so much, Mel. It was excellent and really fantastic stuff. Just seeing those results is just really inspiring. I have a few questions, but maybe I'll just ask one to start with. With your crossing the blood-brain barrier, lots of you're achieving, have you got any idea what that is doing? Is it a bit of an EPR effect, or is it?
Look, that's a good question. So we work very closely with someone called Chris Thurek, who's up at EQ, and Chris has done some really elegant studies because the blood-brain barrier can be compromised when there's a brain tumour. But apparently, according to Chris's work, it's size dependent. So when the tumours are smaller, you don't get any compromising of the blood-brain barrier. But when they get larger, you start getting compromising. These are not very large tumours, so we haven't done the work to see exactly. There are things we could do to try and see if it's been compromised. But given that the zebrafish model suggested that because they're small enough, we think that that's why they're getting through, because there's evidence, around the 20 nanometer mark, they should be able to get through. The zebrafish model suggested to us that they're getting through the endothelial layer. There's been a very recent paper, and I don't know if it's got anything to do with this, because it's only a very recent paper from Warren Chan's lab to suggest that some nanoparticles actually go through cells, right? And rather than just, it's not all EPR. There are studies also to show that the EPR effect is active in some tumours, but not all. We don't know exactly yet. We need to do more work because we've only recently started doing the orthotopic model, but David Ziegler, the clinician that we do a lot of work with, is an expert in DIPG, and he has all the brain tumour models in our institute. So, we'll talk to him about how, I think that his perspective as a clinician is, I don't care how it gets in as long as it gets in and works. But, scientists, we like to know because we want to know how to manipulate it.
Actually, that leads to my question.
Yeah.
And of course, it's about clinical translation. As you know, in cancer with nanomedicine, particularly clinical translation is the element in the room. Of course. So could you share your comments and perspectives on that particular challenge? Yeah,
look, I think it is really... I think it is very challenging. I think we were very excited when we saw that, that first siRNA delivery system. But, there are quite a few other simple delivery systems like formulations and stuff that are in the clinic already. But they're very simple nanoparticles. My feeling is, I think we're in a new era of being able to assess materials. For those chemists in the room who do basic biology, I've got to say, when I first started working in the multidisciplinary teams, we were horrified by the basic biology that was being done to assess these and say, this is really good, this is working, this is not toxic. And I think people are now realising that it's really critical that, you have the right model systems to be testing, and the right, you're using the right sort of technique and asking the right questions. And I think the field has evolved. Unfortunately, we're going to have to negate some of the negativity that's come out from some of the work. There's been a few damning papers, although I do have a few concerns about some of the conclusions that were reached from some of those papers. So I think we're in a new era. I think, I don't know if you saw, we published as part of our Centre of Excellence, the bi-nano standards, the minimum standards, because it's very hard for people to reproduce what other people are doing, because they don't, well, you've got the chemistry issues and the engineering issues, but you've also got the biology, the types of biology experiments are being done. So trying to get people to agree that some principles of standards that should be used so people can try and reproduce work. Sorry, it's a long-winded answer, but I think it's actually quite an important question.
Thanks, Maria, that was great. So your idea of having tumoroids, patient-derived tumoroids for therapeutic profiling, what's the time frame? So if there was a biopsy, what's the the fastest that you could say this is going to be the optimum treatment regime for this particular patient?
That's a good question. We haven't tested that yet. They are doing, so in our Zero Childhood Cancer program, they also using the hanging drop method to try and engross them. But one of the challenges we have is that you don't get a lot of samples sometimes. It's very limited, particularly if it's biopsy, it's very limited. If it's been like taken out, then, and you've got a chunk of tumour, that's a bit different. Usually, let's just say, my idea of this is if you're doing a single drug analysis and you had enough tumour sample, you grow your spheroids. Usually they will grow relatively fast. When they're with them in contact. But let's just say that was 7 days, and then you do your drug treatments for another 48 to 72 hours. I think within 2 to 3 weeks you should be able to do that. But ultimately, patients are not treated with a single drug. It is combination. So in the clinical trial that's going on with the Zero Childhood Cancer Program, the testing that they've been doing so far is single agent. The next clinical trial is going to be combination therapies, both for the trial for the patients in terms of what treatment they get, but also what's going to be happening also in the laboratories as well. So I look, roughly, it's relatively fast. Yeah.
I think we have time for one more question. Okay.
Fantastic work. I was wondering, sort of a point that you raised at the end related back to the tumours and the tumours and the spheroids. Do you think that when you put the tumours in, they're multi-cell types, aren't they? Do you think there's a way that you can expand the in vitro side of things, spheroids, to actually learn more about how the tumours kind of grow and metastasize and are there new avenues for therapeutic?
Yeah, no, look, that's a really good work. So I just showed you, so what I showed you is really the platform more, so we can get reproducibility, screen, different drugs and stuff like that. So nice, simple system. But we actually have Gen 2, Gen 3 inks now, which are addressing other types of questions. How do the tumour cells grow and spread in there? With our Gen 2 ink, we've actually got, we can get some migration invasion in those as well, but we're still optimizing it. But in those cases, we could take the patient, we ask the biology questions as well, and try and understand what happens. Can we inhibit different types of cell types within that tumour, etc. It probably would be a little bit easier. It's not that you can't do it with this, but they're not embedded as in a biological matrix or a biological mimic matrix. They're there as an environment where we can reproducibly grow them. Because if you do the hanging drop method, you do get a lot of variation in the size of the spheroids. So it's hard to compare, these spheroids with those spheroids. Does that answer your question? Yeah.
Okay. Thank you very much. Can you please join me and thank Maria for this great presentation? The live lunch will be provided. It's actually outside, hopefully. And to thank Maria, actually, we take it as a small group Maria for lunch. But I mean, we have Maria there for networking for about 20 minutes.